The main advantage of this technique is that it is very simple, requires only standard equipment, and benefits from the primary advantage of antibodies, which is their specificity. The protocol was originally introduced by Michele Matteoli and colleagues and has proven highly effective. I've used it to test whether a certain protein called ROCD is localized to active synapses.
Begin this procedure by culturing primary hippocampal cells on cover slips in a 24-well plate for 3 days, as described in the text protocol. Pre-warm the reduced serum medium, cell culture medium, and distilled water to 37 degrees Celsius in the water bath. Working under the laminar flow hood to ensure sterile working conditions, prepare the transfection mix in a 1.5 milliliter microcentrifuge tube. First, mix 7.5 microliters of 2 molar calcium chloride with 4 micrograms of each endotoxin-free DNA. Add water to reach a total volume of 60 microliters in a 1.5 milliliter microcentrifuge tube. Now, add 60 microliters of transfection buffer to the mix. To obtain the best results, add the transfection buffer dropwise while shaking the DNA mixture gently on the vortex. Incubate the resulting transfection mix at room temperature for 20 minutes. Avoid shaking the tube during the incubation time. Under the laminar flow hood, remove the cell culture medium from the wells using a 1000 microliter pipette, store it in a separate container, and immediately add 500 microliters